If you had the chance to clone your beloved pets, would you do it? Cloning is defined as a complex process that lets one exactly copy the genetic or inherited traits of an animal, also known as the donor. One of the most common techniques used in animal cloning is somatic cell nuclear transfer, in which the nucleus of a somatic cell is transferred to the cytoplasm of an egg that has had its own nucleus removed. Once the nucleus of the somatic cell is inside the egg, it is reprogrammed by the egg's cytoplasmic factors, which causes it to become a zygote nucleus. The egg is then able to develop to the blastocyst stage, where a culture of embryonic stem cells can be created from the inner cell mass of the blastocyst. The first mammal to ever be created using somatic cell nuclear transfer was Dolly the sheep in 1996. Somatic cell nuclear transfer is often used in the following two types of cloning: reproductive cloning, which produces copies of whole animals, and therapeutic cloning, which produces embryonic stem cells that can be used to create tissues to replace injured or diseased tissues. In reproductive cloning, researchers remove a mature somatic cell, such as a skin cell, from the animal they want to copy, and then they transfer the DNA of the donor animal's somatic cell into an egg cell that has had its own DNA-containing nucleus removed. Researchers can add the DNA from the somatic cell into the empty egg in two different ways. In the first method, the researchers remove the DNA-containing nucleus of the somatic cell with a needle and then inject it into the empty egg. And in the second method, the researchers use an electrical current to fuse the entire somatic cell with the empty egg. Both of these processes allow the egg to develop into an early-stage embryo while in the test tube before being implanted into the womb of the adult female animal. The result of reproductive cloning includes the adult female giving birth to a clone, which is an animal that has the same genetic makeup as the animal that donated the somatic cell. In some cases of reproductive cloning, the use of a surrogate mother may be required in order to allow the cloned embryo to develop. Therapeutic cloning is the creation of a cloned embryo for the sole purpose of producing embryonic stem cells with the same DNA as the donor cell. The most useful and important source of embryonic stem cells is the tissue formed during the first 5 days after the egg has started to divide. This stage of development is called the blastocyst, and it is when the embryo consists of a cluster of about 100 cells that can become any type of cell. Stem cells are harvested from the cloned embryo during this blastocyst stage, which results in the destruction of the embryo while it is still in the test tube. During the development of embryos, they differentiate into stem cells and eventually into differentiated cells. The determination of cell fate is largely achieved by activating some genes while suppressing others through epigenetic modifications such as DNA methylation, histone modification, genomic imprinting, and X chromosome inactivation. These inherited changes in gene expression without alterations in genomic DNA sequences occur during the progression from a still developing fertilized egg to a differentiated embryo, and they also play a key role in the development of somatic cell nuclear transfer embryos. Somatic cell nuclear transfer tends to generate a low cloning efficiency, abnormal embryo phenotypes, and a low viability of animals, which are all believed to be caused by the incomplete reprogramming of donor nuclei. These low efficiency rates can actually be increased by making epigenetic changes in relation to DNA methylation, histone modification, genomic imprinting, and X chromosome inactivation during the process of somatic cell nuclear transfer. In somatic cell nuclear transfer embryos, there is a high DNA methylation status of a pluripotency-related gene called OCT4 and a low level of a tissue-specific gene called THY1. Treatment with the methyltransferase inhibitor and the interference of DNMTs have both been used to reduce the higher DNA methylation level, which effectively improves the efficiency of some somatic cell nuclear transfer. In somatic cell nuclear transfer embryos, there tends to be a low level of both histone acetylation and H3K4ME3, as well as a high level of histone methylation. The overexpression of demethylase or the downregulation of methyltransferase have both been able to improve the efficiency of somatic cell nuclear transfer. In somatic cell nuclear transfer embryos, the loss of H3K27ME3 disrupts post-implantation development. The tissues of animals created through somatic cell nuclear transfer show abnormalities that have been proven to be caused by the abnormal expression of imprinted genes. In comparison to normal reproductive cattle, dead cloned cattle showed the following: highly expressed H19 in the bladder, brain, heart, and lung, highly expressed IGF2 in the heart, kidney, lung, and spleen, and highly expressed IGF2R in the bladder and brain. This means that the DNA methylation of H19 and IGF2 is not being maintained during genomic imprinting for the somatic cell nuclear transfer process, Reforming the abnormal imprinting pattern of somatic cell nuclear transfer embryos may be a promising strategy to improve the efficiency of somatic cell nuclear transfer. In male and female somatic cell nuclear transfer derived mouse embryos, the X inactive specific transcript expression was abnormally high, which led to the inactivation of almost all X chromosomes. This means that DNA methylation of the X inactive specific transcript is not fully established in female cloned embryos of somatic cell nuclear transfer. The deletion or downregulation of the X inactive specific transcript resulted in a 10-fold increase in the birth rates of cloned offspring in mice. Other studies support that revising the X inactive specific transcript in the pre-implantation of somatic cell nuclear transfer embryos has long-term effects on their developmental and postnatal growth. There have been a variety of mammals cloned through procedures that use somatic cell nuclear transfer, and some of the most notable include sheep, cows, dogs, pigs, and monkeys. Reproductive cloning may allow researchers to make copies of animals with the potential benefits for the fields of medicine and agriculture. Researchers have actually been able to clone sheep that have been genetically modified to produce milk that contains a human protein that is essential for blood clotting. Cloned animals can also be used for the testing of new drugs and treatment strategies. The main advantage of using these cloned animals for drug testing is that they are all genetically identical, which causes their responses to the drugs to be uniform rather than inconsistent as seen in animals with different genetic makeups. Animal cloning can also be used to rebuild populations of endangered or even possibly extinct species of animals. Embryonic stem cells from therapeutic cloning have the unique ability to generate nearly all types of cells in an organism, and researchers hope to use these to grow healthy tissues that can be used to replace injured or diseased tissues. Researchers may also be able to learn more about the molecular causes of disease by studying the embryonic stem cell lines from the cloned embryos that have been obtained from the cells of animals with different diseases. The differentiated tissues derived from embryonic stem cells are also excellent tools to test for new therapeutic drugs. Animal cloned embryos can also be turned into something called a stem cell factory, which can be utilized by scientists by turning the stem cells into a variety of things, such as nerve cells that can be used to fix a damaged spinal cord, or insulin-making cells that can be used to treat diabetes. Animal clones also allow farmers to upgrade the quality of their herds by providing more copies of their best animals with the most desirable naturally occurring traits such as resistance to disease, high milk production, and quality of meat production. These animal clones are the ones used for conventional breeding, and their sexually reproduced offsprings are the ones who become the food-producing animals. In conclusion, animal cloning can be achieved through a number of different processes that all produce a genetically identical copy of the original animal or donor. Animal cloning is most notably done through somatic cell nuclear transfer, which is often used in both reproductive and therapeutic cloning. The efficiency rate of clones created through somatic cell nuclear transfer does tend to be relatively low. However, it can be increased by making epigenetic changes in relation to DNA methylation, histone modification, genomic imprinting, and X chromosome inactivation. Animal cloning truly is something that could greatly advance our world by bringing in amazing benefits such as new drugs and treatments, better food production and quality, the replacement of injured or diseased tissues, and so much more. Thank you.